This is a talk about what sort of support as a service there is for analysis of ancient DNA for research community in Sweden. So I'm going to give an introductory presentation. We're also going to get presentation from Tom Martin, who is a project coordinator at the National Genomics Infrastructure. So of course, as you've seen today, and as you will see during the day, there's many reasons why to study ancient DNA. Basically, whenever there is an interest in sort of the temporal aspect of genetics, how was things in the past, how have it influenced the present, and how can you use it to extrapolate into the future? Looking at human evolution, admixture, ecology, evolution, human migration, development of agriculture, structure of prehistoric societies, history of pathogens, and of course analyzing actual and phenotypic traits to the extent it's possible. But there are many challenges to working with ancient DNA, making this technology not easy to access directly, to set up your own labs and your own routines to do so. It has been becoming cheaper with new technology, but still that's put it outside of the realm of possibilities for many researchers, at least until recently. The problem why you need this sort of special procedure, of course, is that DNA is a fragile molecule. It does degrade over time. It becomes shorter and shorter. How fast that is, it depends on environmental conditions. DNA also gets modified over time, so the actual information in the sequence does change. That is, as we have heard, is actually a blessing in disguise, because we can then go to the DNA and actually look at it. Does it have this sort of telltale patterns or chemical modification, which is an indication of it being bona fide ancient. Another problem is, of course, the presence of non-endogenous DNA in the sample, so that's not a contamination, it's part of how the sample is like. And another severe issue, of course, is actual contamination of DNA, ancient DNA, with modern DNA in the lab, which either sort of precludes valuable analysis or makes you misunderstand what analysis actually is being, or what data you actually have. So the way to work with ancient DNA is, of course, to have know-how and experience in selecting material that has a high likelihood of actually containing DNA. So select quality material to sort of, just spend your resources wisely. Use protective equipment for the staff to avoid contamination. Decontaminate the material to sort of reduce the amount of non-endogenous DNA. To have clean rooms for the lab, with positive pressure, so you keep, contemporary dust out. Have airlocks for accessing the labs, not moving between labs and transferring DNA. Having specialized laboratory procedures adapted to this type of material. And of course, validation of ancient DNA, coming back to this chemical modification I talked to earlier. So it is quite a setup to actually do this. We are very fortunate to have very strong research environments in Sweden with labs for this. And historically, lots of research has been done either in these groups or through collaborators with these groups. But with interest growing, it's a limitation of access through research collaboration. And that is why there is an increased focus on infrastructure support for ancient DNA research. So where there is service laboratories that do not engage in projects in order to have publications themselves. They don't have specific research questions, but they're just interested in supporting other researchers. So in Sweden, we have mainly 3 organizations supporting this. We have the Ancient DNA Unit, which I'm head of. We have the National Genomics Infrastructure, and we have the National Bioinformatics Infrastructure Sweden. So the Ancient DNA Unit, we have specialized laboratories, so we can help with sampling and so on. NGI does various types of analysis of RNA and protein, also some data analysis, and at large scale. And National Bioinformatics Infrastructure Sweden, they can provide specialized data analysis for these types of questions. But then, of course, all these 3 infrastructures is part of SciLifeLab. You see our poster of science here everywhere. Many of you are sort of aware of it, using it, but what is actually SciLifeLab? It's a direct government initiative to support life sciences. It has a wide range of different types of units and services. I think more than 40 units currently, 3 of which I mentioned in the last slide. So it's a government-appointed mission to provide research infrastructure in this field. And it's highly distributed. You can find probably various types of SciLifeLab units at all major universities in Sweden, specializing on basically where, depending on what knowledge there is at that particular given research environment. But the Ancient DNA unit is based at these strong Ancient DNA research environments here in Sweden, at the Department of Organismal Biology and at the Center for Palaeogenetics here in Stockholm. The Ancient DNA unit is also part of a wider infrastructure. We're part of the Hyperion Heritage Science, which is an EU-funded infrastructure, and the Swedish Digital Archaeology Infrastructure, which is a VR infrastructure to support data management and data handling of archaeological data, including ancient DNA data. So what can we actually do from the perspective of Ancient DNA unit? So of course, an important part is engaging in project planning, assisting with a discussion about how can an ancient DNA analysis support whatever research a researcher is engaging in. We can help with sampling in clean room, potentially also on-site. We can perform DNA extraction from various types of material in this clean room environment. We can process this DNA to make it suitable for DNA sequencing. We can check those DNA libraries before actually sending them for sequencing. And then we can assist with DNA data production. But we actually don't do it ourselves. We have an excellent facility, which Tom Martin will talk about, the National Genomics Infrastructure, who actually does the DNA sequencing. We can then retrieve the data from NGI, and then we can do various types of sort of standardized analysis. For example, the ancient DNA content, sex chromosome, maternal and paternal ancestry. We can look at close kinship. We can look into microorganisms in the sample, and to some degree, it's also custom analysis, depending on our expertise and capacity. But also here, it was the bioinformatics platform, the National Bioinformatics Infrastructure Sweden, comes in and have a much greater capacity and experience and can provide a lot more support for data analysis. A little bit on ongoing development. This is a fast-moving field, ancient DNA research. So as I mentioned, we're looking into making data available for researchers in a bigger context through digital archaeology infrastructure. We have development projects for increasing our knowledge and ability to support analysis of ancient plant DNA. Further developing microbial and pathogen analysis, and also looking into sediment analysis, we think is a really major field in the future when it comes to ancient DNA. So with that, I'll leave the word to Tom, who's going to talk a little bit about the infrastructure for DNA, RNA, and to some degree also protein analysis.
Okay, thank you, Magnus. So we've heard some fantastic talks today about ancient DNA and DNA sequence. But how do you actually get that DNA sequence? That's where we can help you. So I'm from NGI, which is the National Genomics Infrastructure, and we provide access to technology for large-scale genome sequencing and genotyping, and we also provide bioinformatics support. So we provide access to this large genome sequencing through our large technology platforms that we have. We have a huge DNA sequencing capabilities for both long read and short read. I guess short read is a bit more your thing in this meeting. We have 5 NovaSeq platforms at the moment. We're expanding constantly, so we will soon be acquiring the NovaSeq X, which is the new system. So we are producing more and more sequencing data at a lower cost for you. And NGI is based in Uppsala and Stockholm. So we're 2 nodes. So you can contact us at NGI, and then we can decide, or we can help you find which node will be able to help you best. So we can direct you to Stockholm or Uppsala. So how does that work? Well, first of all, when you get in contact with us, you can fix a meeting if needed. So we can talk to you and try and understand your question. And we can also involve NBIS, which Magnus talked a little bit about, and we'll talk more about after my talk. The next step is you have your DNA and we need to get it into the sequencer. Now, we can't just put it straight into the sequencer. We need to make libraries from it. And we basically stick little adapters onto your DNA, and then we can put it into our sequencing machines. You can do that yourselves, and you can give us your libraries. You can talk to Magnus at the Ancient DNA facility. They can help you with all that. Or if you want to, you can give your DNA directly to us, and we can make the libraries for you. The next step is the sequencing. And this is where our fantastic sequencing platforms that we have can help you. So we sequence your data there at NGI. When your sequence data is done, we will run a QC on it, and our bioinformaticians will do all the demultiplexing and everything so you get your sequences back. And this will be a good time to talk to either Ancient DNA or NBIS to help you to decide what you actually need to do with your data. So how do you get in contact with us? Well, we have a website there, and you also have an email. So you can drop us a line, ask us any questions. We're here to help you. I'll just put a nice little picture of some of the things we sequenced. Yeah. We also have a little stand outside, so come and talk to us. We'd love to hear your questions and interesting projects.
Thank you, Tom. So then I just finish off with a little bit of information about the National Bioinformatics Infrastructure Sweden. So this is also a large operation similar to NDI with lots of capacity, a very wide diversity of experience. So they can provide support with data analysis in various ways. They have sort of short, free discussions on your project, no sort of hands-on work. But then they also have this fee for service where you get a highly discounted analysis, but you pay by the hour. And you basically apply for a number of hours, and then you're approved. The other option, they have long-term support. So this is a review-based project. So then you apply for resource, and a few projects are selected, and then you get a lot of support, typically 500 hours over 1 to 2 years. So that, of course, it's a massive amount of support. It's very valuable. It's also very important that you connect with the NBIS experts, so you actually transfer knowledge to your research team or yourself for doing this analysis. So it's not so just performing analysis, but actually transferring the knowledge how to do this. And also the option of partner projects where you can sort of hire a bioinformatician to work in your team under various conditions. So please, you can reach out to NBIS for discussion these different options, what they mean and what sort of projects they can support. There's also these drop-in sessions that NBIS arranges, very valuable, so you can meet them, and just discuss whatever projects you are interested in doing, and getting hands-on support with various types of solutions to your analysis needs. So with that, I would just like to thank for the time to talk about how we can support research. And as Tom mentioned, we're going to be outside in the coffee break. So maybe the easiest way to get in touch with us is just to come and talk to us. Otherwise, find us online, send us an email. We are happy to help. Thank you.